Bismillah ar-Rahman ar-Rahim. I'm Dr. Mahawagdi, Professor of Histology and Cell Biology. Today we'll discuss histology of the eye. The eye is a complex photosensitive organ. It's responsible for the sense of sight. It's just like the digital camera, but we have to know that the optical system of the eye is highly complex, elaborate in comparison to the digital camera. You know that the eyeball, diameter of the eyeball is about 24 millimeter, about 1 inch, but this inch is the most precious inch in our body. When we take a sagittal section through the eyeball, we can see that it is covered with 3 tunics or 3 layers. From outside inward, we have the outer coat. This outer coat is called the fibrous tunic or the fibrous coat. It's formed from cornea anteriorly, and the rest of the coat is the sclera. While the middle coat or the middle layer, this is the vascular and the pigmented layer. It is called uvea or uveal tract. Uveal tract is formed of 3 main parts: the iris, the ciliary body, and the choroid. It's marked in red. While the inner coat or the inner layer, this is the retina, okay? And this is the sensitive part or the sensitive coat, photosensitive coat. We can see also here spaces. We can see chamber called anterior chamber between the cornea anteriorly and the iris posteriorly, and another chamber we call it posterior chamber. It's between the iris and the lens, and both are filled with aqueous humor, while posteriorly we can find a chamber which is filled with vitreous, and it's called vitreous cavity. Today we'll discuss the coats of the eye, okay, the 3 coats of the eye, and in the next lecture we'll continue the coat, the inner coat of the eye, the retina, together with the accessory structures of the eyeball, which is accessory structures of the eye, including the eyelids, the conjunctiva, and the lacrimal gland. And you know that the eyeball is present in a bony cavity which is called orbit. Let's start with the first coat, the outer coat, which is called the fibrous coat. As I said, it's formed from the cornea anteriorly. This is the most outer part. Here, the cornea constitute 1/6 of the outer coat, while the rest 5/6 is the sclera. What about the histology of the cornea? We'll find that histological layers of the cornea includes 5 layers. This is from outside inward. So this is facing the outside, outer, while the most inner is facing the aqueous humor in the anterior chamber. What are these layers? First of all, the corneal epithelium. Then underneath you will find the Bowman's membrane, and then the corneal stroma or the substantia propria, and this constitutes about 90% of the thickness of the cornea. Then we'll find the Descemet's membrane. This is the Descemet's membrane, and the most inner layer of the cornea, as I said, facing the aqueous humor in the anterior chamber, is called corneal endothelium. So we have a corneal epithelium outside and a corneal endothelium inside. In between, we'll find Bowman's membrane, Descemet's membrane, and the thickest layer of the cornea in the middle is the corneal stroma. So this is a connective tissue we call substantia propria. Let's discuss layer by layer. First of all, corneal epithelium. As we know, any epithelium exposed to the outside must be stratified epithelium. Okay, so this type of epithelium is non-keratinized stratified squamous epithelium. We know that these are multiple layers of cells above each other. The most superficial is the squamous cells. Okay, but this non-keratinized stratified squamous epithelium is a unique type. There are special characters in it which makes it can adapt the function of the cornea. First of all, you have to know that this is the most transparent structure. It's a transparent structure of the eye, transparent structure of the eye. Why? Because there are several factors. We'll mention it in our lecture after we discuss the histology of the cornea. So again, what about the non-keratinized stratified squamous epithelium? It's unique in, look at the basement membrane. This basement membrane is not corrugated as in any basement membrane of the stratified squamous epithelium. It's a straight basement membrane, as you see. Why? Every structure has the explanation to adapt the function. This means that this type of epithelium helps in transparency of the cornea. The cornea is transparent, so it's regular. This is a straight basement membrane, no corrugation of the basement membrane. This is number one. And there are 5 to 6 layers resting on this straight basement membrane. There is a special character to the basal layer of cells and also special characters to the apical layer of cells. You know that the basal layer of cell, this is the regenerative layer. So we have to know that. There is, you can notice mitotic figures. You know what's meant by mitotic figures, seeing the chromosomes in mitosis. Okay, this denotes activity. This denotes regenerative capacity. So the cornea has high regenerative capacity. Why? Because the basal layer can divide and regenerate and repair any abrasion to the epithelium. What about the apical layer? Here we can see the cells, the stratified squamous epithelium, non-keratinized stratified squamous epithelium, okay, with the straight basement membrane. Looking at the basal layer, as we said, the basal layer has high mitotic activity. What about the superficial layer? Superficial layer, as you see, it's flattened cells, squamous cells, flattened cells with flat nuclei, but we can notice here the presence of microvilli. The presence of the microvilli, this is why. There is explanation for this, because the microvilli protrude into the precorneal tear film. What's meant by this? There is a film of tears all the time in front of your eye, in front of the cornea. There is tears here, and these tears will be maintained on the surface of the cornea by this microvillus border. So they protrude into the precorneal in front of the cornea tear film. So this is a modification in the squamous cells of the superficial layer of the cornea. We have to know that the corneal epithelium is rich in free nerve endings, so it's very sensitive. You see here the free nerve endings ramify in between the epithelial cells of the cornea. Then we move to the second layer under the epithelium. This is called the Bowman's membrane. Bowman's membrane. This is the outer layer of the corneal stroma. So from where it comes? It comes from the stroma. Is outer layer of the stroma, formed of collagen fibers. As I said, the stroma, this is a connective tissue containing collagen, and so the collagen fibrils on the outer surface of the stroma will constitute the Bowman's membrane. This is an important membrane. Why? Because it is a protective barrier against trauma and bacterial invasion. So the Bowman's membrane is a barrier against trauma and bacterial invasion. But this membrane does not regenerate It hasn't the capacity to regenerate. And so, if this membrane is destroyed, if the injury involves this membrane, this means that there is no regeneration of the epithelium, and this will leave corneal opacity. Okay, so it's important. If the injury does not involve the Bowman's membrane, it's okay. There will be good regeneration. But if it involves the Bowman's membrane, and this Bowman's membrane is interrupted or destroyed, okay, it will not regenerate and leaves corneal opacity. Then we move to the substantia propria. Okay, this is the third layer. This is the stroma, which will constitute about 90% of the thickness. Most of the thickness of the cornea is formed by this layer. It's formed of 60 regular lamellae. Okay, parallel collagen bundle. You have to know that it's formed of collagen bundles parallel to each other. But if you see by electron microscope, you will find that these collagen bundles are oriented at right angle to each other. So in the form of transverse, longitudinal, transverse, or longitudinal, with uniform spacing in between the bundles. Okay? So as we know, the connective tissue is formed of fibers, cells, and ground substance. We are interested in the collagen, in the fibers. They are regularly arranged lamellae of collagen, of parallel collagen bundles. As we see from the EM, it's at right angle with uniform spacing between the layers. There is ground substance in between these fibers, but the most important is the presence of cells among the collagen bundles. These are fibroblasts. You see, it's just like the fibroblasts, they are fibroblast cells, but here we call these cells corneal corpuscles. According to the cornea, they are corneal corpuscles, but actually they are fibroblasts. So the stroma will constitute of, as you can see, the collagen bundles, and in between the corneal corpuscles and ground substance in between. And these are the lamellae at right angle to each other with uniform spacing. This uniform spacing and the regular arrangement of the cornea will contribute to the corneal transparency. The most important factor here, there is no blood vessels in the cornea, and you have to know that the cornea is avascular. There is no blood vessels, and again, this is one of the criteria of transparency of factors which contribute to the transparency of the cornea: absence of blood vessels or lymphatics. Then we'll go to layer number 4, which is Descemet's membrane. Here the Descemet's membrane, you can see the Descemet's membrane here, and then the most inner layer is the corneal endothelium. What's the Descemet membrane? They said that this Descemet's membrane is the basal lamina of endothelium. So the last layer, the most inner layer, is the endothelium. Its basal lamina will form the Descemet's membrane. What about the corneal endothelium? Endothelium is involved in metabolic exchange between the cornea and aqueous humor. I said that the most inner layer, which is the endothelium, corneal endothelium, is facing the aqueous humor. There is exchange of material in between, and this is done through the endothelium. Now, after we discussed the transparent structure in front of your eye, which is the cornea, okay, we have to know what are the factors affecting corneal transparency. Why the cornea is transparent structure? What are these factors? First of all, corneal epithelium with few layers and straight basement membrane. This is one of the factors. As I said, this is a straight basement membrane and few layers of epithelial cells. Most important is avascular. There is no blood vessels or lymphatics in the cornea. So from where the cornea gets its nourishment, it's either through diffusion from the aqueous humor, as I said, from the endothelium, or from the sclera through the corneoscleral junction, diffusion of nutrients to the cornea. From adjacent structure will help to nourish the cornea. But there is no blood vessels or lymphatic inside the cornea. Again, what about the stroma? There is very regular arrangement of the collagen fibers with uniform spacing between the parallel fibers. You know, it's bundles of fibers. In between, the spacing in between is uniform. And also the lamellae are oriented at right angle to each other. There is other factors, but these are the main factors for corneal transparency. We finished now the most anterior part, the part of the cornea, which constitute 1/6 of the circumference of the outer coat. Now we'll discuss the posterior 5/6, which is the sclera. The sclera is the white of the eye. If you look at your eye, you will find that there is a transparent structure here covering the color of your eye, which is the iris. The color of your eye, this is contributing by the iris, and this is the transparent structure, the cornea, and in between the cornea and the iris there is anterior chamber filled with aqueous. Okay, while the rest of the eye, you can see the white of the eye. The white of the eye, this is the sclera. It forms the posterior 5/6, and it consists of dense connective tissue fibers. Okay, that's why it is white in color. It's formed of dense connective tissue fibers, and these fibers are not regular as in case of cornea. No, they are running in various directions. It's formed of type 1 collagen with elastic fibers. You can see here that this is the sclera, this is the choroidal middle layer, and the most inner layer is the retina. So you have to know that the sclera, the inner surface of the sclera will face the middle layer, which is the choroid. There's changes occurring at the limbus. What is this limbus? The limbus is the junction between the cornea, this is the cornea, transparent cornea, and the sclera, the white of the eye. What will happen at this limbus? And this limbus encircle the eyeball. These changes involved the following changes. One, about the epithelium. Now we discussed the epithelium of the cornea. We said that it's an unkeratinized stratified squamous epithelium resting on a straight basement membrane. Then what will happen? We'll find another structure called conjunctiva. What's this conjunctiva? Conjunctiva, this is a mucous membrane which is covering the front of your eye, starting from the corneoscleral junction. So the conjunctiva is not covering the cornea. This is very important. Cornea exposed directly to the outside, while the sclera is covered with the cornea. So the front of your eye covered with conjunctiva. So the sclera covered with conjunctiva in front of your eye, we call it bulbar conjunctiva. While in case of the conjunctiva lining the upper with lower eyelids, this is called palpebral conjunctiva. Again, what are the changes occurring at the limbus? The epithelium of the cornea, the regular basement membrane, the straight basement membrane, now will continue as the bulbar conjunctiva. I said the bulbar conjunctiva, this is mucous membrane which covers the sclera. So the epithelium will continue with non-keratinized stratified squamous epithelium of the bulbar conjunctiva, which covers the sclera of the eye in front of your eye. What about the second layer? The second layer is called the Bowman's membrane. You know, under the epithelium there is Bowman's membrane. This Bowman membrane will stop abruptly. Why will stop abruptly? Because it will be replaced by subconjunctival connective tissue. Okay? So now the epithelium and the Bowman's membrane replaced by epithelium of the conjunctiva covering the sclera. Which part of the conjunctiva? We call it bulbar conjunctiva, which covers the sclera in the front of the eye, and the Bowman's membrane will stop and replaced by subconjunctival connective tissue, as you can see. Number 3, what about the corneal stroma? This is the corneal stroma, which forms 90%. It will continue as the connective tissue of the sclera, and we know that the connective tissue of the sclera is formed of irregular collagenous bundles. That's why it appears white in color. It's not so regular here as in the cornea. They are irregular in orientation of the collagen fibers, so they are not transparent here. They are white in color while they were transparent in the cornea. Number 4. Again, this is the cornea, and this is the conjunctiva covering the sclera. Epithelium, continue with the epithelium of the conjunctiva. Bowman's membrane will end, replaced by subconjunctival connective tissue. Stroma, regular stroma, continues as the irregular collagen bundles running in the sclera, the white of the eye. What about the 2 layers? The 2 layers, Descemet's membrane and endothelium. Descemet's membrane will continue, and the corneal endothelium, both of them will be reflected on the front of the iris and will be continuous with a discontinuous layer of fibroblast and melanocytes on the anterior surface of the iris. You see here, this is the Descemet's membrane, and the endothelium will be continuous with the iris, the anterior surface of the iris, okay? Color of our eye, okay? The iris will continue as discontinuous layer of fibroblast and melanocyte on the anterior surface of the iris. This is the events which will occur at the limbus. We'll know later in the lecture that this site, limbus, is a very important landmark for drainage of what? Drainage of the aqueous humor present here, okay? The drainage is 3 channels occurring here at the limbus. So the limbus is a very important landmark for drainage of the aqueous. We'll discuss it in the steps of drainage of the aqueous later on in the lecture. Now we finish the outer coat and we'll move to the middle coat of the eye. Middle coat of the eye is the vascular coat. Vascular and pigmented, okay? It's called uvea or uveal tract. Starting from with first structure, which is the choroid, the middle layer, the choroid. There are 3 parts in the uvea: iris anteriorly, ciliary body, and then we'll continue as the choroid. Here is the choroid in the middle. What we have to know about the choroid? We have to know that it's a highly vascular pigmented. Remember these 2 important words, vascular and pigment. So it's rich in blood vessels and there is melanin pigment. As you can see, it's pigmented and vascular. What's the function of this layer? The function of this layer is to support and give nutrition to the retina. Okay, so the blood vessels here is a source of nutrition to the retina, and also the melanin absorbs the extra light. So any extra light passing through to the retina to stimulate the retina, the rest of this extra light will be absorbed by the pigments present in the choroid. So it's simple to know that the middle coat main function is nutrition and pigmentation for absorption of extra light. This is for the choroid. We have 2 other structure. You can see here, this is the layers of the retina, okay? And this is the most inner layer, the retina, and under it the middle coat, which is the choroid, and we can see the blood vessels and the pigments or the melanin pigments. Then to the outside we can see the sclera. Then we'll discuss the second part of the uveal tract, the ciliary body. And the ciliary body, you see that this is the iris and this is the ciliary body. It's just like a triangle, okay? From its position and borders, we can define it as a triangle. What are the sides of this triangle? We have a base here facing the anterior chamber. Here, this is the cornea, this is the iris, and in between the anterior chamber containing the aqueous. Here is at the limbus, as we said, between the cornea and the sclera. We have the ciliary body, here continuation of the iris. It has a base which is facing the anterior chamber. It has 2 long sides, one of it facing the sclera, and the other side is the part of the ciliary body from which ciliary processes will extend. You see this ciliary processes, okay? which will give attachment to the suspensory ligaments of the iris or of the lens, or we call it the zonular fibers. Again, what are these structures? Here, this is the cornea. This is the sclera, and the sclera is covered with conjunctiva. The middle layer is the starting uvea with the iris, then the ciliary body, just like a triangle, as I said. A base facing anterior chamber, a side, long side facing the sclera, and the other side which will face, you have to see in it the ciliary processes, which will give zonular fibers to keep the lens in position. What are the histological characteristic features of the ciliary body? We have ciliary epithelium. So this ciliary body is composed of three main parts: ciliary epithelium, ciliary stroma, and ciliary muscles. This is the ciliary epithelium. This is ciliary body with the ciliary processes. It's covered with epithelium. So it's covering here ciliary body, and in the ciliary, mainly it's on the ciliary processes. It's non-pigmented layer and pigmented layer. What's meant by this? Ciliary epithelium is formed of two layers of epithelium. One of which this epithelium is non-pigmented, the other one is pigmented. Let's see a diagram for it to see the non-pigmented and the pigmented. Again, this is the ciliary body and the ciliary processes. The ciliary processes here is covered with the ciliary epithelium. We'll find it's formed of two layers. An inner layer non-pigmented facing the posterior chamber, and an outer layer pigmented epithelium resting on the stroma of the ciliary body. Why we call it inner and why we call this outer? Because inner is facing the inner part of the eyeball, while outer because it is towards the outer part of the eyeball. Another magnification for the ciliary epithelium, we can see this is the ciliary processes covered with ciliary epithelium. It's not a single layer. There are two layers of ciliary epithelium. One, as you can see, non-pigmented. This is the inner layer because it's facing to the inside of the eye. It's facing the posterior chamber, while the outer layer of the ciliary epithelium is pigmented, as you can see. This is resting on what? Resting on the stroma, on the connective tissue. So this is the outer layer. Again, let's imagine this is the ciliary epithelium, okay, on the ciliary processes formed of two layers, non-pigmented layer and pigmented layer. The non-pigmented is the inner, okay? The pigmented is the outer, facing the stroma of the ciliary body. This is the first layer, the ciliary epithelium. What's under the ciliary epithelium? We can see here the ciliary stroma, the connective tissue, which has the two characteristic features of the uvea as a whole. It's vascular and pigmented. So we can find pigments, melanin pigments, together with blood vessels. Then, what is the third component of the ciliary body? Is the ciliary muscles. So these are ciliary muscles which help in accommodation for near vision and for far vision. What else? It also helps in drainage of the aqueous. You know that the drainage of the aqueous, which will occur at the limbus, and we'll discuss now the steps of drainage of the aqueous. But the three main components again: ciliary epithelium, non-pigmented, which is the inner layer, pigmented, which is the outer layer. Then they are resting on ciliary stroma, which is vascular and pigmented, and we can find ciliary muscle, which are important for drainage of the aqueous and for accommodation to near and far vision. Another histological section to show the ciliary body, the ciliary processes, and to see the ciliary epithelium with the inner non-pigmented and the outer pigmented. What's the functions of this ciliary epithelium? This ciliary epithelium secrete the aqueous. So the secretion of the aqueous is through the ciliary epithelium. Which type? It's here the ciliary epithelium is responsible for the secretion of the aqueous and also for formation of zonular fibers. What are the functions of the ciliary epithelium? The ciliary epithelium, one of its functions is secretion of aqueous. So it's responsible for the secretion of aqueous. Also, it's responsible for formation of zonular fibers of the lens. We know that extending from this ciliary processes, the zonular fibers or the suspensory ligament which will hold the lens in position. So the ciliary epithelium is responsible for formation of these zonular fibers. What else? It forms blood-aqueous barrier. You know there are certain barriers in our body. Two barriers are present in the eye. One is blood-aqueous barrier and the other one is the blood-retinal barrier. Here there is a barrier between the aqueous here and the blood present here through the junctional complexes, tiny junctions between the apices of the non-pigmented cell. So the inner layer, this, which is the non-pigmented cell, this is the non-pigmented epithelium. There is junctions between the apex of these cells together, forming a barrier between the aqueous here, present here, and the blood vessels present in the stroma of the ciliary body. This is called blood-aqueous barrier. So these are the three functions of the ciliary epithelium. This is the ciliary epithelium, inner non-pigmented and outer pigmented, resting on the stroma. This non-pigmented is responsible for the blood-aqueous barrier by tight junctions between the apices of these cells. The third component of the vascular coat or the uvea is the iris. The iris is the color of our eyes. As I said, here we have the cornea externally. There is a chamber between the cornea, the transparent structure, and the iris. This is the anterior chamber filled with the aqueous humor. So the iris is responsible for the color of the eye. This is the iris. It is a disc-shaped diaphragm. As we can see, it's a disc-shaped diaphragm. This is part of it. It's disc-shaped. That separates the anterior from the posterior chamber because it's present here. In front of it, between it and the cornea, we will find the anterior chamber. Behind it, between the iris and the lens, we'll find the posterior chamber. So it separates the anterior from the posterior chambers of the eye. It lies partially anterior to the lens. So it's in front of the lens, but leaving uncovered part of the lens. Part of the lens is not covered. This is the pupil. So this is the opening in the center of the iris. You can see behind it the lens. Again, what is the structure of the iris? It's responsible for the color of the eye. It's disc-shaped, okay? Separates the anterior chamber from the posterior chamber, but it's not complete. There is an opening. It's opening in the center. This is the pupil, and so it lies partially anterior to the lens, leaving uncovered part. So this uncovered part, which is the pupil. Again, this is the iris. You see the pupil in the center and the iris here, and behind it we can find the lens, and in between the iris and the lens we'll find the posterior chamber. Histologically, again we have anterior surface of the iris, we have stroma, and we have posterior surface. The anterior surface, if you remember, I said that the dismal membrane and the endothelial cells of the cornea is reflected to discontinuous or continuous with the discontinuous layer of fibroblast and melanocyte on the anterior surface of the iris. This is part of the iris. This is the anterior surface of the iris. Posterior surface pigmented, and in between you'll find the stroma. A stroma, this is responsible for the color of the eye because it contains the pigments, okay? And this is vascular, stroma is vascular, containing muscles. Mainly you have two types, constrictor papillae muscle and dilator muscles, which will control the diameter of the pupil. This is the iris, anterior surface, posterior surface. This is the lens, and in between the iris and the lens, here this is the posterior chamber. Here is the pupil. Again, cornea, anteriorly. Here is the limbus between the cornea and the sclera. Corneoscleral junction is the limbus, and from a middle coat we can see the iris here, diaphragm, which is penetrated by, or in the middle there is a pupil, so it covers the lens except for the part anterior to the part facing the pupil. We can see the iris, the ciliary body, and the rest is the sclera. What about circulation of aqueous? We know that formation of aqueous starts from the ciliary epithelium of the, yes, these are the ciliary epithelium. The ciliary epithelium of the ciliary body will form the aqueous, and then this aqueous will circulate. It will pass from the posterior chamber through the opening between the lens and through the pupil, between the lens and the iris. Here is the iris and this is the lens. It will pass here through the pupil to communicate, to pass from the posterior chamber into the anterior chamber between the cornea and the iris. And then we'll go to the drainage. Where is the site of drainage of this aqueous? Because the aqueous is recirculating. It's formed and drained, okay? Where, this is what I said about the limbus, which is the landmark here. There is opening called trabecular meshwork. From this trabecular meshwork, yes, trabecular meshwork here, we'll go to a canal called Schlemm, canal of Schlemm, which is a circular, irregular opening encircling the limbus around the eyeball, which is endothelial lined vascular space. So it's a vascular space lined by endothelium called canal of Schlemm. There is drainage from trabecular meshwork, this opening into the canal of Schlemm, and then will be drained to the episcleral vein. So again, what are the steps of the aqueous circulation or circulation of the aqueous humor? First, it's produced by the ciliary epithelium and then passes from posterior chamber through the pupil into the anterior chamber in between the iris and the lens. At the limbus, it will go at the limbus, will pass through trabecular meshwork. These are the trabecular meshwork, okay? And then exit through the canal of Schlemm. As I said, irregular canal encircling the eyeball at the limbus, okay, which is endothelial lined. It's lined by endothelial vascular space lined by endothelium, and finally the drainage will be to the episcleral veins. This is the pathway of the aqueous humor circulation. This show you the trabecular meshwork and the canal of Schlemm, and then to the episcleral veins. So the eye limbus, again, this is the transition between the cornea and the sclera at this site, and it's a vascular zone. And you can see here as the canal of Schlemm, it's a regular circular channel running around the limbus. We said its role in drainage. Pointing again to the canal of Schlemm. As long as the secretion, rate of secretion and rate of drainage is of normal, okay, in the normal range, this is a normal situation. But if there is increased secretion or there is diminished or any problem in the drainage of this aqueous, this will lead to increased intraocular pressure, and this clinical condition is called glaucoma. So there is balance between secretion and drainage, but if the rate of secretion or increased or the rate of drainage is decreased, this will lead to clinical condition called glaucoma. Okay, thank you so much.